Okay, group 4. Who is going to be the co-host?
I'll be the co-host.
Okay. Just a moment. So you can share your PowerPoint first.
Okay.
We'll just wait for 1 more minute and then we can start. Okay, you can start now.
Okay. Good morning, everyone. We're group 4, and today we're going to share with you our study on marijuana. Our presentation consists of the following parts, including some background knowledge, the negative impacts of marijuana and its medical usage. In the Q&A session following our presentation, we would like to hear your questions if you have any, which will help us better understand. Our labor division is mostly in accordance with our strength and expected effect of our presentation. The 3 boys took charge of information collection, and they will be responsible for the Q&A session later. Yujing and I finished designing the slides, and we will be presenting all the contents. First of all, what is marijuana? It is also known as cannabis, a psychoactive drug extracted from the cannabis plant, indigenous to Central Asia and the Indian subcontinent. The use of its psychoactive effect can date back to at least 3000 BC in Iran. It was brought to the Western Europe in the early 19th century and began to be criminalized throughout the 19th and 20th century. But more recently, some regions start to legalize its recreational use, such as Canada in 2018. This diagram shows the legal status of recreational marijuana consumption. We can see that marijuana in most areas around the world is still illegal. Marijuana has 2 main ingredients, the tetrahydrocannabinol and the cannabidiol. These 2 only differ in one functional group, but they have distinct functions, which will be discussed later. The cannabis works via a special system, which is called the endocannabinoid system. This is a unique communication system in the brain and body that affects many important functions, including how a person feels, moves, and reacts. Different from normal neuronal communication, the endocannabinoid system functions through retrograde signaling. That is, the neurotransmitter is released from the postsynaptic neuron to the presynaptic neuron. They can control what happens next when these cells are activated, limiting the amount of neurotransmitter that gets released, which in turn affects how messages are sent, received, and processed by the cell. There are 2 types of receptors in the endocannabinoid system, the CB1 receptor and CB2 receptors. CB here stands for cannabinoid. The CB1 receptor appear in both the central nervous system and the peripheral nervous system, while the majority of them are located in the hippocampus and amygdala. The CB1 receptor is actually a GPCR protein which downregulates the cyclin AMP, and its functions include psychoactive, neuromodulatory, and analgesic effects. The CB2 receptor can be found in immune cells, lymphoid systems, and peripheral nerve terminals. It has immodulatory effects rather than psychoactive effects, and it helps control the immune function and central nervous system by inhibiting the inflammatory signaling pathways to return the body to a state of homeostasis. And now, my teammate Yu Jing will introduce some detailed mechanisms and negative impacts of marijuana.
If asked for the main reason why they use this largely illicit drug, the majority of cannabis users in the world will probably answer, "It relaxes me." So during marijuana smoking, THC triggers the brain to release large amounts of dopamine. It is what gives us a pleasant high. THC, one of the two major components in marijuana, works on both CB1 and CB2 receptors. To healthy people without the drug's intake, endocannabinoids add to CB1 and 2 as key regulators of the synaptic transmission and plasticity. They are synthesized on demand following physiological or pathological stimuli. But the brain will gradually adapt to the increasing amounts of marijuana. The brain adapts to this large amount by reducing production of neurotransmitters or reducing its sensitivity to its own endocannabinoid transmitters. It is understandable that people can't synthesize enough endocannabinoids enough after quitting from marijuana smoking. And smoking marijuana can also heighten our sensory perception and our perception of time. In the hippocampus, THC changes the way we process information, so our judgment can be impaired. And the hippocampus is also responsible for memory forming, so it may also be difficult for the marijuana takers to form new memories when they are high. There are also studies showing the negative effect of THC exposure in different ages, using rats as the model organism. Studies in rats also shows the adolescent exposure to THC is associated with an altered reward system, increasing the likelihood that an animal will self-administrate other drugs when given an opportunity. And that's why marijuana is usually thought as the gatekeeping drugs. Another major component in marijuana, CBD, has been shown to act as a negative allosteric modulator of the CB1 receptor. CBD is a negative allosteric modulator of CB1 receptor, preventing THC binding, stimulation, and its psychotropic effects. So how does the endocannabinoid systems work in our neural systems and affect us? Let's take these CB1 receptors induce NMDA receptor-dependent long-term potentiation as an example. The endocannabinoid-mediated neuron synapse plasticity is much more diverse than the conventionally anticipated that to induce the long-term depression of presynaptic neurotransmitter release. As shown in the picture, red dots represent glutamate. Endocannabinoids released from the postsynaptic dendrites to bind to the CB1 receptors could inhibit the glutamate release. However, endocannabinoids can also act on astrocytes to increase the release of astrocyte glutamate, which modulates synaptic transmission and plasticity by activating glutamate receptor or NMDA receptor. And the D-serine release could also be triggered by endocannabinoids activating CB1. When the occupancy of the NMDA receptor binding site of the D-serine increases to a threshold, the downstream signaling pathway that underlies long-term potentiation induction will be triggered. So when people take marijuana, THC and CBD as exogenous and cannabinoids also work in such a complicated systems that can have stimulatory or inhibitory effects. So the underlying mechanisms of marijuana-induced syndrome still require further research. And now please let me pass our time to Dan Yi to let her talk about the medical usage of marijuana.
Okay, back to me again. Although marijuana is more commonly used as drugs and toxins, they actually can serve as medicines. We will briefly introduce a few methods where they can be used, but the detailed mechanism is unclear yet. The Epidiolex is an FDA-approved medicine, which is actually highly concentrated cannabidiol. CBD can be used to treat seizures associated with two types of epilepsy, the LGS and DS, and it functions via CB1 or CB2 receptor. There are 3 FDA-approved drugs containing the 2 different synthetic forms of tetrahydrocannabinol. The first form is dronabinol, which can be used to treat nausea and vomiting associated with cancer chemotherapy, and the loss of appetite in people with AIDS. The other one is nab, with similar usage to dronabinol, and it functions via the CB1 receptor. It is proposed that THC can be used to treat cancer pain as well by reducing the functional connectivity between the amygdala and primary sensory motor area, since the amygdala activity contributes to the dissociated effect of cannabis on pain perception. Other than psychoactive effect, the scientists are trying to treat cancer with THC. The tetrahydrocannabinol can inhibit tumor angiogenesis and metastasis, as well as triggering apoptosis and inhibiting proliferation of the tumor cells. However, because THC can also induce suppression of the anti-tumor immune response, so it is still questioned whether THC can be used to treat cancer. That's the end of our presentation, and now we're open to questions. Please feel free to unmute yourself. Thank you.
Okay, great. Question time. Come on, feel free to ask the questions.
Hi guys. I just want to ask, what are some of the health, like what kind of damage would it do to your health if you smoke marijuana for a long period of time? Is there any
Hi.
long-term health effects?
Yeah, thank you for the question. So basically, the harm that marijuana did is not as strong as some other harder drug, like probably like heroin or something. But still, there are evidence showing that if you take a very large amount of marijuana and like you take it for quite a long time, you actually can have we call it marijuana use disorder. And I think there's a survey saying like 30% of marijuana users actually have this kind of problem, like marijuana use disorder. And in some severe case, they can still have like some sort of addiction, like cannot discontinue using this drug. And also it can affect your create anxiety and influence your brain function, something like this. Yeah.
Okay, any more?
Yeah, I have one, so maybe I'll just throw my question. Do you know about the presence of this thing called cannabinoid food, meaning they infuse cannabis into food like tea and milk tea and all this stuff, cocktail, right? So would the effect of cannabis be as pronounced as in the case of infused cannabinoid food, and would the heat kind of degrade them, or how much should you take when you, like the concentration, the toxic doses, right? Because if you infuse into food, you wouldn't know how much you're going to infuse it in the first place. So do you think there should be regulation for this as well?
So you mean the marijuana like intake through oral ingestion, right?
Yeah, put it in tea, put it in cocktail, right?
Okay, yeah.
It's a new trend.
Yes, it's true that marijuana is usually taken by smoking, but there are also many like edible products, like you mentioned. Actually, the difference is that so firstly, smoking have faster effect, but edible product have slower effect, and also I think it's like edible product can have. I mean, it's not like edible product that always have stronger or like more potent effect. for psychoactive, because it depends on like how much you add, and actually there are different form of marijuana. Like some people take it through dry plant product, and some people take it through oil, like oil extracted from cannabis. So it's not like edible product is always more potent or like more harmful than inhalation, like through smoking or vaporization. So yeah, if that helps.
Yeah, sure.
Okay. So any more questions?
Oh, okay, overdose on cannabis. So it's not, I mean, I think there's another survey saying like actually people die on marijuana use is actually less than alcohol use, right?
I'll give more detail on this question. It is not easy to overdose in this marijuana. The LD50 of marijuana is quite large, and generally people use marijuana by smoking or eating. So in this way, it is almost impossible to overdose and die, unless you do injection very pure marijuana, but it's very rare, very rare.
Okay, good. I think yes. If no more questions, then we can end here. Okay, thank you. So just a moment. Okay. So group 5, who is going to be the co-host?
Yeah, I'll be the co-host. Wrong button, and there we go. Yeah.
Okay, you can start.
Okay. Hello everyone. Nicotine. Probably everyone in this Zoom meeting have heard about it as one of the most notorious substance which exists in the cigarettes. With no doubt, it's poisoning our health. But every coin has two sides. It could be beneficial in certain aspects, and I will explain to you why, and briefly introduce its background information. In fact, nicotine occurs throughout the tobacco plant and especially in the leaf naturally. But until 1571, the crude nicotine was known, and by 1828, the compound was finally obtained and purified. For its name, nicotine, it actually comes from a French ambassador, Jean Nicot, who is the first person who sent the tobacco seed to Paris in 1550. And like I said, every coin has two sides. The reason why I would say it could be beneficial to mankind is that scientists is working on nicotine therapy, which could increase alertness, improve coordination, and enhance cognitive performance. It could be used for those who have cognitive issues, such as patients suffering from Alzheimer's disease. It was once used as pesticide since 1980s, but it's banned by the EU on 2009 due to its highly toxicity. But on the other hand, it's much notorious for an addictive component in tobacco, since the dependency could develop with continuous usage, which means it's hard to get rid of it once you're obsessed with nicotine. And it also increased the blood pressure, heart rate, and blood flow, which contribute to the hardening of arterial wall, and it may cause heart stroke. In the following slides, my group will introduce its basic feature and the scientific reaction. Please, Garrison.
Can you guys hear me?
Yes.
Okay. Thank you, Nikki. Let me introduce the structure and appearance of nicotine. Nicotine is a bicyclic compound with a pyridine ring, which is indicated with the red arrow, and a pyrrolidine ring, which is indicated with the blue arrow. And in room condition, nicotine exists as a colorless to pale yellow or even brown oily liquid. It colors depends on the exposure to air and or light. Also, it also available as a powder. Now let's talk about the pathway to the brain. Usually, nicotine is inhaled into our lungs when smoking cigarette, then diffuse into our blood in the air sac, and then enter the brain along with the blood circulatory system, and then reach the target site with the nicotinic receptor by passing through the blood-brain barrier. Then let's talk about the degradation and excretion of nicotine. Nicotine is rapidly metabolized in the liver to continent, which exhibit an elimination half-life of around 2 hours, which the structure of continent is shown on the top right-hand corner. And then the rate of nicotine metabolism is determined primarily by the enzymes in liver called CYP2A6, which the structure is shown on the bottom right corner. And the excretion of nicotine is mainly through urine in the forms of continent and its derivative. Now let's pass my time to my groupmate Stone.
Okay, can you hear me?
Yes.
Okay. So the main target of the nicotine is the nicotinic acetylcholine receptor, and it's one of the most widespread neuroreceptors, and it can be found at many places like the PNS, skeletal muscles, and many parts of the CNS. So these receptors are found mostly on the post-synaptic side, but also some on the presynaptic side for feedback systems. And there are two types of these receptors. The neuronal type it can be found in many parts of CNS and on the postganglionic side of the ANS, and the muscular type that forms the neuromuscular junction. And it's considered as a fast receptor because it's quite sensitive to agonists and antagonists. I would like to mention that adrenal medulla is the only organ that has this receptor on it and has a big role on how nicotine affects our body. And we're going to talk about it in the next session. So nicotinic acetylcholine receptor is an inotropic receptor, and it's bound to the membrane. Each receptor is made up of 5 subunits, which consists of 17 subtypes. And as you can see, the consisting subunits of the muscular type doesn't have much variations, but more combinations can form for neuronal types, especially for those in the brain. And the key family is the alpha subunits. It makes up the ligand binding site with other subunits. And the different combinations can change the properties of the receptor for different functions. Next slide, yes. The natural agonist of this receptor is, of course, acetylcholine, and other agonists include nicotine, choline, and epipetamin. And typical of this receptor is not very selective, and it's quite permeable to sodium and potassium ions. And some receptors in the brain with specific combinations is also permeable to calcium ions. So in a normal situation, the agonist binds to the alpha subunit of this receptor located on the postsynaptic side, which increases the permeability of sodium and potassium so that sodium would rush in and potassium rush out of the cell, causing depolarization, then EPSP, and contribute to action potential.
Thank you, Stone. So now I'll talk a bit more about how nicotine is related to addiction. So in a broader sense, nicotine actually upregulates the excitatory neurotransmitter in our brain, which is glutamate, and it downregulates GABA, the inhibitory neurotransmitter. What this gives rise to is the increase in dopamine release. And it also has a direct effect on, well, as Stone mentioned, on the nicotinic acetylcholine receptors, which stimulates the dopaminergic neurons to release dopamine directly. And as we all know, dopamine gives off pleasurable effect. It's also critical for positive reinforcing effects. And these dopaminergic neurons mainly reside in our mesolimbic system, and which is located in our midbrain. It's also located in the corpus striatum, part of the also part in the midbrain, as well as some of them are located in the frontal cortex. And how nicotine actually causes the addiction is mainly by desensitization of the nicotinic acetylcholine receptor, which is caused by repeated nicotine exposure. And this is done by phosphorylation on the alpha subunits by multiple types of kinases, PKA and PKCs. And what happens after the desensitization of the receptors is that it requires a higher dosage of nicotine to compensate for the same amount of pleasure. And if you withdraw from nicotine, say if you quit smoking, it will cause us withdrawal symptoms, which is mediated by CRF and CRF1 receptor system, which is essentially the stress response system of our body. Next up, Daniel will talk a bit more about how nicotine affects the other parts of our nervous system.
All right, thank you, Owen. So besides the central nervous system, nicotine also affects the autonomous nervous system, right? So if you remember from Stone's slide, nicotinic receptors are also found on the postganglionic neurons on the sympathetic and the parasympathetic pathway, which activates both of them. So it also activates the chromaffin cells of the adrenal medulla. And what this does is that it is activated to release catecholamines, including adrenaline and noradrenaline. So you might ask yourself, if nicotine affects both sympathetic and parasympathetic pathway, wouldn't the effect kind of cancel each other out and thus creating a paradox? Well, the explanation is quite simple, right? So first of all, there is more innervation of sympathetic nerve in the blood vessels, right? And in addition to the contribution of adrenal medulla, the sympathetic effect will be more pronounced and dominant, resulting in a state of relaxed alertness, vasoconstriction, increased blood pressure, all of which are part of the fight or flight response. All right, now let's talk about nicotine's effect on the muscle type nicotinic acetylcholine receptors. So luckily for us, the effect of nicotine, or rather the affinity of nicotine to the muscle type receptors is quite low, because if it is at the same level, it will be actually fatal for humans. But why is that the case? So research has found that in the neuronal type receptors, there is a strong cation-pi interaction between the nitrogen cation of the nicotine and the aromatic side chain of tryptophan 143, also known as TRPB of the alpha subunits. And this interaction is actually quite weak to almost non-existence in the muscle type. And further research has done that shows that the difference is actually caused by a difference of a single amino acid further down the proteins, which is either lysine or glycine, resulting in the opening of this receptor box, allowing different kind of affinity to different kind of receptors. So in conclusion, nicotine is a very addictive compound that activates the nicotinic acetylcholine receptors. And its effects on the body is by acting on the reward system of the CNS and as well as activating the fight or flight response. And fortunately for us, the affinity of nicotine to muscles is low, and as such, really causes any problem there. Thank you, and feel free to shoot your questions.
Okay, question time.
I actually have a question for you guys. So I want to know how does nicotine pass through the blood-brain barrier?
Actually, the nicotine could pass through the blood-brain barrier is due to its relatively small molecular size and its lipid-soluble natures, which is kind of similar to why lipid-soluble molecule could pass through the cell the phospholipid bilayers. Yeah.
Okay, any other questions?
Hello. You mentioned that nicotine could be fatal if the receptors in the muscles are more sensitive. How does that lead to fatality?
Oh, well, imagine if your muscle sensitivity is as high as, let's say, your CNS, all this stuff. It will actually go through like continuous stimulation leading to paralysis of multiple body parts, which might include your heart, right? And as such, it might lead to muscle paralysis and death, which is quite bad actually, or results in a state of tetanus, which it contracts until there is no more contraction because you cannot depolarize it any further, which is quite bad, to say the least. Yeah.
Okay.
Okay, any more? Okay, if no, then thank you, group 5. So
I will be the people to share the screen for group 6.
okay, just a moment. Okay, we can share now.
Okay. So good morning, everyone. So by looking at this picture, can you guess what are we going to talk about today? So yes, we're going to talk about alcohol today. So ethanol is the commonly type of consumed alcohol. So it has the effect of facilitate socialization, but at the same time, it can depress brain function and may cause alcohol dependence. So although we know alcohol do harm to our body, but the reward system in our brain may encourage us to keep consuming alcohol. So under normal condition, a balance exists between inhibitory neurotransmission and excitatory neurotransmission. But after we drink alcohol, it will induce the activity of inhibitory neurotransmitters like GABA, and at the same time, it will inhibit the activity of excitatory neurotransmitter like glutamate. But after a long term of alcohol exposure, our brain will try to restore the equilibrium of the depressant effects of alcohol. So our brain will try to increase the excitatory neurotransmission while decrease the inhibitory neurotransmission. So first of all, let us look into the dopamine pathway of the brain. And we also call this pathway as the reward system. So you can see this part is the VTA where the dopamine produced. And then it will go through the mesolimbic pathways to different brain areas. So when this circuit is triggered, we will feel happy, and then we will be motivated and reinforced to reach the stimuli again. So when we drink alcohol, this is what happened in our brain. First of all, it will increase the opioid peptides level here, and then it will reduce the GABA release in VTA. Then this will trigger the mesolimbic pathway here so that we will release more dopamine. And this will trigger the reward circuit that this will induce the reward circuit what I mentioned just now, and so that we will want to drink more alcohol. But after a long term of alcohol exposure, it will cause the dopaminergic hypofunction, which means a decrease in dopamine synthesis and an increase in dopamine reuptake. This will lead to symptoms like depression and alcohol craving, Parkinson-like symptoms, and anhedonia. And now I would like to pass my time to my group mate to talk about the serotonin pathway.
Okay, so moving on to the serotonin pathway. Serotonin is produced by neurons in our brain and projected to different parts of the brain to control our emotion and the motivation of certain behaviors, such as alcohol abuse. Besides, the binding of serotonin to receptor will initiate various biochemical events that relate to alcohol drinking behaviors. So after acute alcohol exposure, our serotonin level in the nervous system will temporarily increase through the enhanced signal transmission at the serotonergic synapse, triggering more release of serotonin. On the other hand, serotonin is cleared more slowly from the synapse. As the serotonin is known as the happy hormone, with more serotonin, people will feel happier after drinking. Moreover, acute alcohol exposure interferes with the function of serotonin receptors. For example, alcohol targets directly at the 5-HT3 receptors that enhance the electrical signals generated by the receptor. The excessive stimulation of neurons will in turn increase the release of other neurotransmitters that play roles in alcohol intoxication, which gives the symptoms of drowsiness or loss of judgment and memory. Although most people would think drinking makes them relaxing, this might not be the case in the long term. With chronic exposure of alcohol, the serotonin level will decrease as our brain cells produce less and reuptake more serotonin back to the cells, leading to depression. Moreover, excess alcohol leads to a bonding mechanism called tolerance development. That is how our brain cells adapt to changes with re-establishing normal function. So in this case, 5-HT2 receptors will increase and stimulate the serotonin activities at the receptor, resulting in alcohol withdrawal syndrome, such as experiencing increased anxiety level after cessation of drinking. So now I will pass the time to my teammates to talk about the glutamate pathway.
So then let's talk about the glutamate pathway. Glutamate is one of the most important excitatory neurotransmitter, which increase the activities of signal receiving neuron. It affects cells by activating receptor to allow positive charge ions to flow into the cell. So there are several glutamate receptors, but I will mainly talk about NMDA receptor because it's a likely target for alcohol effect. And here is the important structure of NMDA receptor. And then, so when there is an acute alcohol exposure, alcohol will reduce the phosphorylations of a site on the NR2B subunit called Ti-1472. This regulates the functions of NMDARS. Alcohol inhibits the NMDARS by activating a tyrosine phosphatase called STEP. STEP forms a complex with NMDAR and regulates its activity via the phosphorylations on Ti-1472. And this reduces the NMDAR functions and then further reduce the activity of neuron and finally results in the depression effect. So while for chronic exposure, to compensate for chronic alcohol-induced NMDA receptor inhibition, the number of NMDA receptor on the cells along with the levels of the receptor activity will increase. This is achieved by the alcohol's effect on the modulations of the regulatory process, including alternative splicing exon on NR1 and NR2B subunit. So this will increase the gene expression and polypeptide level of these two subunits, and then further cause the excessive NMDA receptor activity, and finally contributes to seizure. Then let's pass to Venus to talk about the GABA pathway.
Let's talk about the GABA pathway. First, GABA is an inhibitory neurotransmitter, and alcohol will act through two receptor subtypes, the GABA A and GABA B. The alcohol will mainly affect the GABA A receptor as agonist. Alcohol will not speed up or involve in the production of GABA. They just regulate the GABA activities. For the acute alcohol exposure, the alcohol increased the GABA activity in presynaptic and postsynaptic side of the neuron. For the presynaptic side, the alcohol acts on the GABA releasing neuron in order to speed up the release of the GABA and also inhibit the reuptake of the GABA. For the postsynaptic side, the alcohol acts on the signal receiving neuron, which they in order to facilitate the activity of GABA receptor, especially the GABA A. Therefore, the people will become lethargy as they have the higher GABA concentration. And regarding to the chronic alcohol exposure, the alcohol will affect the GABA in two aspects. The first, it will affect the gene that encoding the expression of the GABA-A receptor, which in order to increase the alpha-4 subunit and decrease the alpha-1 subunit of the receptor. Moreover, the alcohol will also increase the neuroactive steroid release in order to regulate the GABA-A receptor activities. Therefore, people will get the desensitized of the GABA-A receptor, so the GABA-A receptor will be tolerant to alcohol, so the people will get overdose or alcohol withdrawal symptom afterward. In conclusion, alcohol affects different kind of neurotransmitter and causes various effects. For the acute alcohol exposure, the people will get mood elevation first due to the dopamine increase and will get level G afterward since the GABA increased. Therefore, moreover, the neurotransmitter also influence different parts of our brain as the slide shows here. Let's pass the time to our groupmate to talk about the structural plasticity and treatment.
Speaking of change in structural plasticity, alcohol is proven to cause alteration in areas responsible for learning and memory. And it is further discovered that alcohol has caused structural plasticity change in dendritic spines. The function of dendritic spines are known for controlling the individual synapses and affect their sensitivity to glutamate. They are classified by their length and diameter of the spine head. Of course, there are some external factors which might cause the structural change of dendritic spines, for example, the age and temperature. But it is also shown that the effects of alcohol on dendritic spines in different parts of the brain on different individuals, based on their habits of alcohol consumption and their age. For example, in adults, loss of dendritic spines and reduced length of remaining spines are shown in CA1 hippocampal pyramidal neurons. And for chronic alcohol consumers, there is an increase in stubby and wide spine. An elongation of dendritic spine is observed. And also the change in size is also observed. If adolescents consume alcohol, their spine density increases. For treatment of alcoholism, there are three medicines used widely. Disulfiram is used for inhibiting the activity of ALDH and also adverse accumulation of acetaldehyde. But the side effects are having metallic taste in mouth and patients may experience drowsiness. Naltrexone is used for involving endogenous opiate system and reduces the release of alcohol-induced dopamine. Patients may experience nausea and joint and muscle pain. Acamprosate restores imbalance between excitatory and inhibitory amino acid neurotransmissions. The side effects are gastrointestinal disorders and headaches. And this is the end of our presentation. Now we are open up to Q&A session.
Okay, good. Questioning time. Anyone?
I got a question because I heard in the dopamine pathway, you said the GABA will decrease, but overall the pathway, you said again, the GABA will increase. Does it sound like contradictory or is there anything that I'm missing?
So actually, for this pathway, GABA will release first so that dopamine can be released afterward. Will be reduced first and then dopamine can release afterwards. But actually, for the overall effect, the GABA will increase because alcohol is actually a depressant to our brain system, and this effect is actually different in different individuals. And also the alcohol effect to GABA also affect different brain regions. So it's actually not that contradictory. And when we drink alcohol, we may experience the happiness first and then the drowsiness afterwards. So maybe some research findings have found that when dopamine increases, our brain don't want the happiness last for a very long time. So after that, GABA will also increases.
Okay. So any other questions?
Yeah, I'll maybe shoot in another question. All right. So as a responsible alcohol drinker, right, I was told that not daily, but a weekly intake of a small amount of alcohol does bring a beneficial effect, which I'm not sure if you mentioned it in your presentation. So is there any truth to that, or would that eventually lead to addiction and all this stuff? Because there are research that say that it brings like good, results to you, in alertness and stuff.
Okay.
Like when we do this research, we didn't find any things regarding like alcohol to good to our body, but instead, like for chronic exposure, we find that there are a lot of negative behavioral changes, and when we drink too much alcohol, like increased anxiety, increased risky decision making, and also some increase the chance to abuse different kind of drugs. But I think it may depends on people, because it may depends how good we process or like detoxic the alcohol in our body. Like we prepare another slide, because in our body we have another enzyme called aldehyde dehydrogenase, which will try to convert the toxic acetaldehyde into non-toxic substance. So if you have more enzyme in your body, you may be better cope with alcohol.
I see, that's where all my problems come from. All right, thank you.
Okay, good. Any other questions?
Hello. I also heard that for the chronic misuser of drink alcohol, they are having a high chance of having sudden cardiac death. Could explain why for the long-term alcohol drinker could have such an effect to the body, why they got the high chance of having the cardiac death.
I think I'm not sure about this question, but I think it may because it slows down the body functioning in our body. I'm not sure about that.
It's okay.
Okay.
Isn't that I mean that question? I don't think it's much related to neural heart. It's more about the blood vessels, and it hardens and it changes the blood composition that makes the heart more vulnerable to those things, I think.
Okay, so I think we should stop here. Okay, thank you, group 6. So group 7. Who will be the co-host? Group 7?
We have a co-host.
So you are the co-host, huh?
I'm not the co-host.
You are not the co-host. So group 7.
Make me the co-host.
Okay.
Hello, can you hear me?
Maybe you have to speak up. So you are the co-host.
Can you hear me now?
Okay, you can start to share your PowerPoint. So Helen? Okay, you can start. Okay, ready?
Hey guys, can you all hear? Hello guys, can you guys hear me?
Yes.
Yes, clearly. Oh, sorry for the technical problem.
Okay, we can start now.
Yes.
Good afternoon, we are group 7 and today we're going to talk about LSD. So we'll follow the outline of the presentation and I will first start by the history and origin of LSD. So LSD was first synthesized in 1978 by Swiss scientist Albert Hofmann, and he was working with a chemical found in ergot, which is a fungus that grows naturally on rain and other grains. The invention of LSD was unintentional, so Hofmann didn't discover the hallucinogenic effect of LSD until 1943, when he accidentally ingested a small amount from skin and perceived extraordinary shape with intense play of colors. So 3 days later, Dr. Hoffman decided to test LSD personally with a carefully measured dose. After taking it, he was unable to continue working, so he drove home by bicycle, and that was the first LSD trip in history, and April 19th came to be celebrated as Bicycle Day. During the Cold War, the CIA conducted experiments with LSD for mind control information gathering, but the results were not as good as ideal. And in 1960s, non-medical use of LSD was popularized by intellectuals, poets, and musicians like the Beatles, Watson and Crick, or even Bill Gates and Steve Jobs. In the popular press, LSD became associated with youth-led social movements like anti-war, anti-cultural demonstration, and sexual liberation, which largely ignore the potential drawbacks of its widespread use. So the media began reporting on the negative outcomes associated with LSD use. The cases of young people committing suicide due to LSD abuse accelerated the fear of people to it. And finally, in the 1970s, LSD was officially prohibited and placed in Schedule 1. And now I will pass my time to my fellow groupmate. She would talk about the clinical overview.
Okay, so I will first talk about the duration. Usually, the first effect of the drug when taken orally are felt 30-45 minutes after taking it, peaking at 2 to 4 hours, and used by the intravenous route will produce a much quicker action, usually within 10 minutes. The experience of LSD are lengthy, with the effects of higher doses lasting for 6 to 12 hours, and it may take 24 hours to return to normal. The next are the side effects. Extreme mood changes can occur, and if taken in large enough doses, the drug produces delusions, visual hallucinations, and the user may also experience impaired time perception with distorted perception of the size and shape of objects, color, sound, and their own body image. and sensations may seem to cross over, giving the feeling of hearing colors and seeing sounds. And these changes can be frightening and can cause panic. So some LSD users also experience severe, terrifying thoughts and feelings, like the feeling of death. The physical effects of LSD are unpredictable from person to person. They can include nausea, loss of appetite, increased blood pressure, difficulty sleeping, dry mouth, and tremors. But we know these effects from statistics and data, since the mechanisms are not yet fully understood. And since there's no known lethal dose of LSD, death is often due to a direct injury while under LSD influence. And since when under the influence of LSD, the ability to make sensible judgments and see common dangers is impaired, making the user susceptible to personal injury or death. So I'll pass it on to my groupmate to talk about the mechanisms.
So moving on, before heading on to talk about the mechanism of action, I would like to first give some data with reference to with regards to the pharmacokinetics. So it is oral drug. LSD comes in different forms as shown in the diagram. Something that is worth mentioning is about the half-life. So the half-life means it takes the time taken to require for the concentration of the drug to drop to half of its original concentration. It's take around 4 hours, you can say. But then do not be misled. It is kind of misleading because it seems like it takes very short period of time to metabolize all the drugs, but it is actually not the case, because for the binding of LSD with the receptors, it's actually it has high affinity and it binds to the receptors and it has a long-lasting effect. So sometimes the hallucination, the psychological effect can last up to 12 hours, and some even develop some syndromes that they can still, the hallucination still reoccur. Moving on, this is a structure talking about the LSD. So LSD actually acts as a partial agonist against the receptor. This is the receptor from serotonin, HTL2A, the serotonin receptors, and the pink one is the receptors, and the light, you see the very light blue sticks in the middle, that is LSD. And we also have a 2D chemical structure on the left. So this is some kind of familiar because we learn about the pharmacodynamics. We know about GPCR. So LSD, the receptors, serotonin receptors, the 2A types of 5H of serotonin receptors is actually belonging to the group of GPCR and is coupled to GQ, G protein. So what does the binding? The LSD resembles serotonin 5HT, and it initiates a downward cascade, which leads to an increase in protein phosphorylation and activation in calcium binding proteins. And that's addressed by my groupmates. We're still not entirely certain about the mechanism of LSD because first the studies on central nervous system is still lagging, is still challenging, and then we understand brief mechanisms at the surface level, but then what is required to induce hallucination is still uncertain of. And thanks to the other groups who explained with different kind of addictive drugs, and especially the group that explains addiction, for our drug LSD is actually, when comparing with the other drugs, you can see from the diagram on the left, it has a relatively low potential for physical dependence because it is not heavily involved in dopaminergic signaling. Yes, it binds to serotonin receptors, it also binds to different types of receptors, but it is not heavily involved in dopamine, the GABAergic receptors, and so on and so forth. And that's why it is not quite an addictive drug. And this is the 2D chemical structure. Simply by taking a look at the 2D chemical structure, we cannot simply resemble any similarities. So this is a very recent paper from this year in September 17th, and the scientists elucidate the X-ray crystal structures of the receptors and see what is required for the binding of LSD with the receptors. And they conclude that salt bridge is actually essential between D1553.32 and a positively charged nitrogen in each ligand. For this, it is essential required to make a bonding. And this is some data from the diagrams drawn from the paper. you can there are different types, but then in a short of time we're going to brief it. But then just to point out that part is actually the D1553.32 is actually essential for the binding, and the diagram on the D diagram is actually about the mutation, and they do mutagenesis studies, and somehow this part is very important for all ligands to bind to these receptors. And we also prepare some data with regards to LSD affinity. Just for your information, binding affinity is a kind of dissociation constant, so a low level indicates that the complex is going to dissociate to a very low extent. So it indicates reflects strong binding and it has a long-lasting effect that I just mentioned. So moving on, I'll have David to explain the therapeutic value of LSD in terms of the other neurological disorder.
Now I'm going to talk about the therapeutic potential of LSD. Scientists found out that LSD is a potential drug for treating neurological disorders. Here I will discuss two hypotheses of how LSD treats depression. First, LSD can treat depression by affecting the default mode network in the brain. The default mode network is a loose connection of different brain areas and neurons that communicate together to give a normal, everyday consciousness when we are at rest. For example, self-reflection, which is the evaluation and introspection of past events and experience about oneself. People with depression seems to have a hyperactive default mode network. This patient will give automatic thought loops that are inaccurate and excessively negative. Studies show that LSD significantly chips away at organization within the default mode network. This will cause a temporary loss of consciousness and eventually stop the rigid thoughts and prevent excessive rumination. Second, LSD can treat depression by promoting structural neuroplasticity. Studies show that LSD can promote neuroid outgrowth by robustly promoting a large increase in both the number and the total length of dendritic branches of prefrontal cortical neurons. LSD also increases both the density of dendritic spins and synapses in prefrontal cortical neurons. All these are promoting structural neuroplasticity, since dendritic atrophy and the loss of dendritic spins in the prefrontal cortex are hallmarks of depression. The structural plasticity that's constructed by LSD seems to be a potential treatment of depression. This may link to the GABAergic pathway because reduced GABAergic activity in the prefrontal cortex has been linked to depression. Promoting structural plasticity in the prefrontal cortex may increase GABAergic activity and therefore treating depression. And now I will pass to Losha to talk about the research and future development of LSD.
The use of LSD was prohibited in the early 1970s due to the lack of molecular biology research approaches and also because of the social stigma attached to LSD at that time. In the past 14 years, no studies of LSD have been conducted in humans until very recently. Luckily, the promise and potential of LSD are still there. After 14 years of technical advances in brain imaging and molecular neuroscience, it enabled us to build on more solid work regarding LSD. With the help of new technologies, it allowed the researchers to effectively link the brain's transformations with mood change, so they are able to better understand that what actually happens inside the brain under the influence of LSD. This data would contribute to further investigations of the therapeutic potential of LSD in psychiatry. For example, by using functional brain imaging, we now know the effect of LSD on neural network. Just like my group mates have said, studies have shown that LSD can decrease default mode network integrity, while increasing default mode network have been described in depressions. So this phenomenon revealed the potential of LSD with antidepressant effects. In addition, future studies should investigate the effects of LSD and its analogs, both short-term and long-term. Many other hallucinogenic drugs might show similar effects with LSD, especially its analogs. This gives us the hint for developing more psychiatry drugs. So more research on physiological and psychological mechanism and the therapeutic potential of hallucinogenic drugs like LSD in psychiatry is needed. This is the end of our presentation. Take it or not take it. This is the answer for you to answer. Thanks.
Okay, so question time.
I do have a question because you mentioned that your drug have a lot of side effect, but at the same time, it's also treating depression. So when the doctors prescribe this drug to patients, how can they how do they minimize the side effect?
I think the positive side of LSD, as I mentioned, that is treating depression, but it's all just the experiment part. Is that the scientists use a very low dose, microdose of LSD to do the experiment and find out that LSD is actually have some benefit in treating depression. So it's not at the clinical trial now.
How about I add on some information, because as our group has mentioned, studies on LSH metabolism or the mechanism is still, we're not entirely certain of how it induced the hallucination. And most of the content that we propose today is the more widely accepted phenomenon observation, but there are still some kinds of hypotheses. And for the therapeutic parts, as David mentioned, it is still under studies. We are trying to understand how it exerts its clinical significance, but then they are not being prescribed today. Yeah, so doctors are not really prescribing drug LSD to the patients with depression because they are still under studies. Does that solve your question?
Yes, thank you.
Oh, you're welcome.
Good. Any other questions? Any other questions? Anyone? Okay, if no more, then we will finish today. But just before you go, I just want to let you know that the assessment sheet, you can give it to me after we finish the project presentation. Okay, you don't need to send it to me now. And just in case that you don't know how to, it's maybe a little bit confusing. So in each, the top 1 to 11 is group 1 to group 11, and you put the numbers there, and then rank their presentation. Is it okay? So you can give it to me after the presentations, maybe even 1 week after. Okay. Okay, so next time we will have a group 8 to group 11 on Wednesday. So pretty good. All of you are doing a good job. Okay, so we'll meet again on Wednesday, and then we'll finish off the group project presentation. Okay.
Could I ask
Yes.
back the question about the alcohol group, about why the cardiac death is not about related to the brain function, because it's about the heart beating, abnormality in the heart beating. It beat faster, so then should be related to the neurology of our brain. So could one answer me? It's not related to the cardiac death. explain why it isn't related, because I don't understand.
Maybe do you mind to give you your email address so that I can do some research and answer you later on?
Sure, but I think it's a guy in your group answer me.
No, actually, that student is not in the group.
Ah.
Yes, he just gave a comment. He is not in her group. Yes.
Oh, sure. I would send you a private email. So
No, you can just put your email in the chat box, and after I get the answer, I can try to answer you.
Thank you so much. Okay. This one and following with the connect USD dot picture.
Okay.
Thank you. Yeah, I'm done.
Okay, good. Yes. Okay, so if no more questions, then we'll meet again on Wednesday. Okay. Okay. Goodbye.